Good afternoon, everyone. That's a pleasure to be here to present my work, as mentioned, which is on the identification of a metabolomic marker of grape infection with Esca complex disease. So Esca, thank you. Esca disease is a fungal disease that is triggered by a combination of different fungi. It is part of the Grapevine trunk disease family, which are specific cryptic disease developing in the plant trunk xylem. They lead to, in general, symptoms like grapevine loss of vigor, as well as eventually the death of the plant. And it is usually identified thanks to specific symptoms leading to those kind of tiger straps aspect on the leaves. But it's not consistently all over the world, as in America it's also known as black missile, as some black dots are observed on the grapes. Unfortunately, we don't have any current treatment efficient against the disease, and up to our knowledge, it was shown that all cultivars have sensibility to the disease. And strangely, for such a disease which is of growing importance, as it has been shown to spread or getting more and more observed everywhere in Europe and other parts of the world, information is still relatively scarce, and we are missing information on the factors that promote the development of the disease, as well as both fungal infection process and plant resistance strategies against this disease. This leads to problematic of our work, which is connected to the fact that Esca disease can develop for many years asymptomatically in the trunk, and when symptoms become apparent, plants are usually already beyond salvation. But another interesting fact is also that those symptoms are not consistent through the year. When it starts, you can see symptoms for many years, and then it stops, and the following one, it will look completely healthy plants, while later on, all of a sudden, they will restart again, symptoms. So this leads to the need to develop a non-destructive way to diagnose Esca at an early stage, the disease, and also to improve our understanding on the Inca disease and the possible factors of either susceptibility or resistance to the disease. So our work focused on the identification of biomarker of infection and defense. So a bit of methodology. We worked in vineyard of Aragonese cultivar, also known as Tempranillo, that were like 17 years old vines, which had been previous to the experiment monitored for 2 consecutive years for different fungal disease. So we selected vines that were consistently asymptomatic and symptomatic, and sampled both grapes and leaves of the different plants at the harvest stage. So samples were then analyzed for metabolomic using different gas chromatography techniques, and we also perform like a transcriptional profiling using qPCR. So, as it's, many fungal species, we first validated the different fungi that were observed, that were present in the plants, and also wanted to confirm that our asymptomatic plants presented the symptoms. So we cut 2 of the harvested samples and separated in different sections. You can see in the middle like specific symptoms of the early disease with a black or dark brown streak. And on the symptomatic, you also see the white throat, which is a typical symptom of a late stage disease when it turns to a much more serious stage. Fungal identification, I'm sorry, I'm not going to pronounce them or even try, confirms that the fungi most commonly found with the Esca disease, as well as, in that case, for multiporia, which is specific of the white throat symptoms. So we perform, as mentioned, like a metabolomic analysis that allowed us to quantify here in the grape berries like 93 compounds. Among them, 81 were identified using the Max Planck Institute library. First, you can see on that the principal component analysis, like the relatively good quality of our samples with good grouping among the different repeats, and obviously a good separation between, we are glad to see a good separation between asymptomatic and symptomatic samples that seem to drive the first dimension of this PCA. Looking at the different variable, or in our case, like the metabolite that drives the separation. Here you can see like the loading trimmed of the unknown compound and which were hidden. The main drivers that seem to separate the symptomatic to asymptomatics were coming from two classes: the fatty acids and the volatile organic compounds. So samples, as mentioned, separated from asymptomatic to symptomatic. And on the second dimension, you can see mostly separation between the different infected samples that seem to be driven from one side by the volatile organic compound and the other one from mostly fatty acid and some lipid components, metabolites, So we process to the same methodological approach on our leaf samples. Here we managed to quantify 139 metabolites, with 79 of them being identified. Once again, we can see like a nice separation between symptomatic and asymptomatic, with one exception. And the separation seemed to be driven mostly by the phenylpropanoid and the sugar. On that case, once again, the second dimension seems to separate samples, different infected samples, and were mostly driven on one side by the amino acids, and on the other one by some flavonoids. Moving on from multivariate analysis, we looked into our samples like metabolite by metabolite. The first take is we interestingly observed some difference between the berries and leaves, with some lipids, terpenoids, and volatile organic compounds that were only detected in the berries, while some flavonoids and sugar conjugates were only detected in leaves. Not going to go through all of the sample individually, metabolites, but yeah, you can see here like some different significant change between asymptomatic and symptomatic samples. Looking more specifically into the fatty acids found only in the grapes, you can see that symptomatic samples are also selected ones, like mostly, higher quantity in comparison with the asymptomatic. And we observe like some of those compounds were either involved in the plant defense, either in signaling or by possessing some antimicrobial, antifungal activity, like the tetracosanoic acids, or other ones were more like very long-chain fatty acids that are found to be component of the waxy cuticle. Here, it's quite interesting because some of another project that we are working on, also on Esca, has shown that berries undergo a certain number of symptoms that seem to be kind of typical of a drought stress. So we postulate on that matter that the change on the cuticle might be to try to limit water evaporation to counter that drought symptoms. And so the result seems to show that the plants mount like a fatty acid-related defense to try to respond to the Esca infection. We are now looking a bit more in detail in the volatile organic compounds. So those are quite interesting and, well, important for the grapevine community. Well, one grapevine, as they are the one responsible mostly for the one flavor and component. You can see here on the individual ones put on the side like a plus or minus to mention if they are used, each component is more associated with either what is perceived as positive or negative flavor in your wine bucket. So here, for example, the 2-transhexenal is associated in general with more like, well, is positively perceived and associated more with green flavors, while the ethyl octanoate is more like on the fruity notes. So yes, you can see here that symptomatic plants, with the exception of the 2-transhexenal, have a higher quantity of those volatile organic compounds, and that most of them are rather bringing like which, well, default or what will be perceived negatively on a wine, like acetic acid, ethanol, or ethyl acetate. So we postulate that symptomatic Esca will lead to grapes, which will, well, the grapes harvested from symptomatic Esca will affect wine quality and flavor bucket. Now we looked, well, after that, to the phenolic compounds. A separate analysis for quantification didn't allow us to measure any significant difference between symptomatic and asymptomatic samples. However, looking at individual compounds, we could see that syringic acid and ursolic acid were present in higher abundance in the symptomatic sample, which is interesting as they have been shown to possess some specific antifungal and antimicrobial activities, which will suggest a reaction to stress. Based on those results, we decided to do like a preliminary transcriptional profilings on some selected genes, so that we identify some gene of interest based on literature from other fungi. And well, as you can see here on 2 of our selected genes, we didn't observe, well, in any of the one we looked, except for tocopherol, any expression difference between our 2 different groups, which, well, now will push us to do like more RNA sequencing approach for a more comprehensive and in-depth analysis. So this leads me to the conclusion of, well, what we have done so far, which as take message is that both grapes and leaves undergo like metabolic changes with the Esca symptom development, and that those changes are organ-specific and observed at a stronger level in the berries. Inside of those berries, you can see that fatty acid and volatile organic compounds are the metabolic class which have the most altered, which have been the most altered in terms of quantity. And well, interestingly, those are compounds which are associated mostly, well, for the fatty acid, like in defense or Esca, well, sorry, plant defense response to Esca, and that might lead as well to, in that case, like a cuticle modification for other symptoms described like, what do you say, limitation, while volatile organic compounds contributor to wine aroma are also modified, which putatively leads to altered wine aroma profile. So we are going to carry on the transcriptomic analysis as mentioned with RNA sequencing, and we managed to identify a certain number of metabolites with strongly altered profile, which we can consider, or we would like to consider as putative biomarker of Esca infection, and which we are going to carry on and validate using different cultivars as well as different vintage. And yes, as a takeover as well, all those data will be made available on a public repository on the Metabolite platform. Any of you is interested to compare with work or do other analysis. So to conclude, I would like to thank people from our group, the Fruit Functional Genomic and Biotechnology group that contributed to this work, as well as Pedro Reich and Cecilia Rego from the ESA, and Alexander Orban and Joachim Kopka from the Max Planck Institute. Of course, the FCT for funding this research and the Integral Cosaction to give me the opportunity to come here. And finally, the GBG committee to giving me the opportunity to present this work. Thank you for your attention.